Okay, welcome to today's colloquium. Our speaker is Professor Nigel Goldenfeld from the University of Illinois. Nigel holds a Swanland Endowed Chair and is a Center for Advanced Study Professor at of Physics at the University of Illinois Urbana-Champaign. He's director of the NASA Astrobiology Institute for Universal Biology at Illinois and leads the Biocomplexity Group at the university's Institute for Genomic Biology. Nigel received his PhD from the University of Cambridge in 1982. He was a postdoctorate fellow at the Institute for Theoretical Physics at the University of California, Santa Barbara. And in 1985, he joined the faculty at the University of Illinois. Nigel has been an Alfred P. Sloan Foundation Fellow, a University Scholar at the University of Illinois, a recipient of the Xerox Award for Research, and a recipient of the A. Nortzik Award for Excellence in Graduate Teaching. In 1996, Nigel co-founded Numerics, a company that specializes in high-performance software for the derivatives marketplace, a little bit different. He serves on the editorial board for several journals, including the Philosophical Transactions of the Royal Society in Physical Biology. Nigel is a fellow of the American Physical Society, a fellow of the American Academy of Arts and Science, and a member of the U.S. National Academy of Science. And today, his talk will be on universal biology, the genetic code, the first billion years of life on Earth. So I think 1 hour should be adequate to cover that topic, in detail. So won't you please welcome our speaker.
So thank you very much for your kind introduction and for inviting me here. I've had a lovely day. It's a pleasure to talk again at this renowned physics institution, and I want to commend you on not studying biology, because biology is messy and unpleasant, and nothing in it makes any sense, and it's certainly not regular or universal, unlike the laws of physics. But on the other hand, I do want to talk about some rare things in biology, universal biology. And so let me first start off by saying why you shouldn't study biology. And the reason is because everything in biology has an exception. Now, when I started doing biology about 15 years ago, I started talking with Carl Woese, who I'll tell you a bit about during this talk. He's a wonderful microbiologist. He was a physicist by training, and he, in fact, was the first person to map out the evolutionary history of life on Earth, and I'll talk about that. And when I told him about this statement, which is everything in biology has an exception, which is what everybody believes, he said, no, that's not true. Everything in biology came through the results of evolution, and that's the unifying principles of biology. And in fact, those are the things that I want to talk about today. So the Institute for Universal Biology has this logo, and I want to explain to you some of the elements of this fanciful design to you. So let's start off with this thing. So how many people in the audience recognize what this thing is, what this is a cartoon of? Anybody? A couple of people do. What do you think it is? A branch of a branch of life, or something like this, a family tree. Species separating out. That's right. This is the sort of tree of life, and I'll show you what that tree of life is. It was first drawn. Well, it was drawn most famously by Darwin, of course, but it was actually measured by Carl Woese at the University of Illinois. So this tree of life is something we're going to talk about, and how it was determined. This thing here is not so familiar to you. This is what happened before the tree of life. So this tree of life really tells you about everything that happened from about 3.8 billion years ago. Okay? So for the purposes of this talk, we really don't care very much about that. What I'm more interested in is this era here before the tree of life started. Before the tree of life, there was in fact life. And what kind of life was it? Well, I'm going to show you that there's very strong evidence that this life was not a tree, but was a network. And the idea of species and even individuals was not well defined at an earlier point in life, earlier point in time. So we're going to talk about this network of life, this phase that you haven't heard about, and that's got a fanciful name, the progenote, but think of it really as life being a network as opposed to a tree. And I'm going to explain to you that in the talk. Now this spiral thing here, well, maybe to you it looks like a spiral galaxy, because I know there's a wonderful astrophysics and cosmology group here, but you'll see that this spiral galaxy actually has some chirality, and for the purposes of this talk, what I'll be interested in is chirality of life's molecules. This spiral galaxy has these, is broken up in this way, and that's meant to represent a kind of genome sequence that you see in DNA, and so this is DNA forming some kind of spiral galaxy. It's universal biology. We're funded by NASA, and so this is the space bit of it, but also it's meant to describe chirality. And I want to talk about all of these things in this talk, mostly about the tree of life and how it emerged from the network of life. I'll talk a little bit at the end about chirality, and if I have time, which I won't, I'll try to talk about the idea of universal biology and what that really means and what one might understand from it. So let's get into the main topic of the talk, and that is the evolution of the genetic code. So this is how I want to this is the window on which I want to peer at early life. Just like in astronomy and cosmology, you peer back in time by looking out into space. So we peer back in time in biology by looking back at genes, and we look at how they are structured and so on. Now, everything in this talk that I'm going to tell you is based on data, a very sophisticated analysis of data, that, as you'll see. And at the end of the talk, there's a little bit where it's more speculative, and I'll tell you at that point where we leave the realms of data and analysis and get into perhaps more speculation. But most of what we're going to talk about is the analysis of the genetic code. So what is the genetic code? So what is this thing here? Anybody? Somebody said the genetic code. It's not the genetic code, okay? Okay, so the newspapers say this, and even scientists sometimes say this. You see this sequence of A, C, Gs, and Ts, and you know that has something to do with DNA bases, but that sequence is your genome, but it's not the genetic code, okay? So let's just talk about what those A, Cs, and Gs, and Ts are, and then I'll tell you what I mean by the genetic code. So A, C, Gs, and Ts stand for these nucleotide bases, and they're paired in this AC GT pairing that Watson and Crick discovered. It's known as Watson-Crick pairs, and from that you construct your DNA, and the sequence of those is the instruction set for your body. So let's have a look at how that actually works. So this is, I'm assuming that nobody here knows any biology, and so let's do this at a very simple level, which is about the level I know of biology too. So you start off with DNA. So DNA is built from 4 nucleotide bases, A, C, Gs, and Ts. Now think of DNA as being the plans for, say, your body or the cell, or maybe even the plans for Fermilab, let's say, okay? So you've got the DNA sequence which has the whole plans for Fermilab embedded in it, and let's suppose, God forbid, that something goes wrong with the projector in this room, and we have to call in an electrician to fix it. The electrician, wanting to fix the wiring of the projector, does not want to see all of the plans of Fermilab. They just want to see the plans for the electrical system in this lecture room. So what you would do is you would just Xerox off a couple of pages from the full Fermilab plan, and then the engineer would go ahead and she would fix the projector. So biology does the same thing. It doesn't have a Xerox machine. It has a thing called RNA polymerase. What RNA polymerase does is find coding regions on DNA. It goes around those and spits off another nucleic acid called messenger RNA, which is also built from 4 nucleotide bases, and you can think of as being just the plans for a gene or plans for a particular protein structure or something like that, and that's the analogous to just Xeroxing the wiring diagram of this lecture room. Okay, so this mRNA is just nucleic acid, but you're not built out of nucleic acids mostly, you're built out of proteins. So how do we get the proteins? Well, there's another machine, a very complex machine called a ribosome, that now goes along the mRNA, and it spits out, it reads the sequence here, and spits out the proteins. Now the proteins are built from 20 amino acids. There's more amino acids in nature, but biology only uses 20 of them. And the question is this, how does it do that? Well, you've got 4 nucleotide bases and you've got 20 amino acids. How do you code for 20 possibilities with only 4 bases? Obviously, if you have just one thing you read, that's not going to be good enough. If you have 2, that's 4 times 4, that's 16, still not enough for 20. So the next thing you can have is triplets. So what the ribosome does is it goes along the messenger RNA, it reads the nucleic acids in groups of 3. Those groups of 3 are called codons, so AUG, ACA, GGG, AAA, and so on. It reads those things, and what it does is, through a process too complicated to explain, it reads those things and then collects the amino acids that correspond to that, sews them together, and produces the protein that's coded by this sequence here. And so these things are called triplet codons, and this process is called translation. Now, it's a very fundamental process in a cell. Obviously, a cell will not work if you don't have this machine in it. So this machine is going to be the central player in everything I'm going to tell you. Now, the genetic code. So we've got this map that is used by the ribosome to translate the message from triplets of 4 bases into 20 amino acids of life. So the genetic code is literally a code. Okay, it's not the sequence of DNA that makes your genes, it's the code book that biology uses. And it's sometimes called the universal genetic code because it's the same genetic code that's used in every organism that we know of on this planet. That's how you can do genetic engineering. You can take a gene from a jellyfish and put it in a pig, and the pig will glow green. Okay? That can only happen if they're using the same code book to translate the DNA, and they do, and so we have a universal genetic code, which I prefer to call the canonical genetic code. So here it is, and here's how you read it. So here's the 4 bases, U or T, U, C, A, and G, standing for those chemical symbols. We don't need to worry about them. Here they are up here, and here they are up here. So you read this row, this column, this row, and then the last one here. So, for example, U, and U, read that off. It's for this amino acid called phenylalanine. And you can do C, U, A, and that would code for leucine, and so on. So that's how you read this thing, and that's called the genetic code. So here's the question. The question I want to ask is this. If we were to reboot life on Earth, okay, some asteroid crashes into the Earth, destroys all life on it, life starts again, would we get the same genetic code? Okay, so we're rebooting life. So how many people in the audience think the answer is no? They would get something completely different. Okay, a few people. I don't know, 5 or 6, 7. How many people think you'd get something the same? Okay, my daughter does. Yeah, that's good. I think you'd get something the same. How many people who said no have heard this talk before, heard my answer, and decided they don't believe it? Okay. Nobody, okay, good, all right. So my answer is you'd get yes, and I want to explain to you why that's the case. So here's what I'm going to tell you. I'm going to tell you that the genetic code is not a frozen accident. Frozen accident is what Francis Crick called the genetic code and why he didn't really work very much on evolution. And here's his reasoning. It's really clever reasoning. He said, look, some organism was the organism from which all life on Earth was descended. We'll call that organism the last universal common ancestor. Okay, that organism had some genetic code. Okay, whatever it has, we must have the same one. And the reason is this, we're all descended from that organism, but let's suppose you change the genetic code. It mutates in some way, like everything does in biology. If it mutates, then you're going to get the wrong proteins. If you get the wrong proteins, then the organism is not going to be very competitive and won't survive. And so therefore, it's impossible for the code to evolve, because any organisms which had an evolved code would not be competitive against the other organisms, and so they would die out. So codes can't evolve, and therefore it must be frozen, and it's an accident because it just happens to be whatever genetic code the original ancestor of all life on Earth had. Okay? So that's not true. Here I'm going to show you it's not frozen, and I'm going to show you it's definitely not an accident. And I'm also going to show you that it has another property, which is that it is optimal in minimizing errors. It's a wonderful, wonderful code. Suppose you gave yourself the homework exercise of doing a bit of intelligent design. You said, I'm going to design a genetic code which has the following properties. Okay, the code is operating in a very noisy environment. It's operating in a cell. The DNA mutates, so you get mutations in the DNA sequence. The ribosome moves along and makes mistakes as it translates and makes the proteins. So how can you guarantee that you're going to get the right proteins that you're supposed to get? What you would do if you were trying to do this intelligently is you would design it so that if there was a mutation and you get the wrong amino acid, the amino acid you get to make the proteins is still pretty close to the amino acid you were supposed to get. And if you could make a code that has that kind of stability, that kind of robustness, then it's going to be much more robust towards errors than a code for which that doesn't work, where if you get the wrong amino acid, you get something totally fatal and the organism dies. I'm going to show you that the genetic code has this property. It's optimal, very close to optimal in minimizing errors. Okay, okay, I'm only telling you what I'm going to tell you yet, so I'll be happy to take a question, but go ahead.
That almost optimal genetic code must have come about after other suboptimal codes were tried.
Precisely, that's what's going to be the subject of my talk, okay? So I'm going to explain to you the mechanisms by which such a code could emerge, okay? Does that answer your question? Yeah, thanks. Okay, so then I'm going to show you how we can simulate the evolution of the genetic code. We're going to have to do that in some coarse-grained way, and I'm going to show you what this means for our interpretation of life on Earth. And then if I have time, I'll talk a little bit about the chiral symmetry breaking in life and a little bit about universal biology. But this is going to be mostly what I'll have time to talk about. Okay, I told you that at the end of this talk, I'm going to get into speculation, but most of the talk is going to be founded in data, the data I just showed you. But let's start off with a fairy tale. The fairy tale is a fairy tale that's told in most of the biology textbooks that you get at say, high school, and the fairy tale goes something like this. The Earth started 4.6 billion years ago. That's completely true. Then what happened is there was some process of chemical evolution that nobody really understands. Molecules became more and more complex. Maybe there was an RNA world. Maybe there was a world where there was no DNA, but still some kind of information processing. Nobody quite knows what happened then. But at some time, the first organisms emerged. And those organisms were very, very simple unicellular organisms, which are called prokaryotes, and then they gradually became more and more complex until there was another thing that happened, and then you've got multicellular eukaryotic organisms that emerged. Eukaryotes are the organisms that have cells with a nucleus in them, and prokaryotes are cells which are simpler. They're just a big membrane, and inside of that is the DNA and a bunch of chemicals. So the idea was you start off life here, something happened, life started, you get something simple, then you get something more complicated, and these things are called prokaryotes because they sort of came before the eukaryotes. So that's a fairy tale. Everything I just told you is either wrong or not, or there's no scientific evidence for. Okay, the person who did the most to show that to be the case is Carl Woese, shown here in characteristic pose at his desk on November the 3rd, 1977, on the front page of the New York Times, no less. So what had he done to be out here? Well, here's the headline. It's wrong, of course, because it's a newspaper. But it says, scientists discover a form of life that predates higher organisms. And what he discovered was not just a new species, like every now and then you'll learn that somebody found a new butterfly or a lizard or a kind of bird sitting in their garden or something like that. He discovered a whole domain of life, a whole classification of life that had previously not even been suspected, and it was right there underneath our noses all the time, and he discovered it, and that's what made him famous. So he died a few years ago, but of course everything in this talk was work I did in collaboration with him. Now, how is it that he was able to make this remarkable discovery, and what did he actually discover? So he had the idea, he was a physicist, he didn't know much about biology in terms of looking at creatures and looking at counting how many legs they have, what color their feathers are, and stuff like that. So he said, look, if life evolves, then we are going to see the evolution reflected in the sequence of the DNA. So let's take a part of the DNA that codes for something that is guaranteed to be in every cell. And that thing, of course, is the ribosome. You have to have a ribosome in cell, in fact you have many of them, in order that you can do translation. So he looked at a fragment of a gene, technically called 16S ribosomal RNA gene, and he looked at it for different species, and so here it is, human, yeast, corn, E. coli, and various other organisms like this. These sequences look pretty similar, but there are small differences. And those differences reflect the mutations and the divergent evolutionary history of life. So what you can do then, if you're given all these sequences, just like looking at particle tracks at Fermilab or LHC or anywhere you like, you put the things together to try to see what order they come in, and what was the, in this case, what is the minimum path, the edit path between the sequences. That gives you the evolutionary history of the organisms. And there's technical ways to do that. You can look for sequences that are similar in some entropic sense, or maximum likelihood, or parsimony, or various other metrics, and people now know how to do this pretty reliably. So here's what he found. Here's a sort of a very simplified roadmap of it. So this is these are bacteria, which you've heard of. These are eukaryotes, which you've just heard about. There's you, okay, just there. Virtually everything else you see on this tree, and many of the things that you can't see, are microbial. Most of life is microbial. We are just a blip on life. We could just be wiped out tomorrow. It would make no difference to life on Earth. Okay? Now this is the thing that he got famous for. He discovered this third domain of life, 1, 2, 3, this one here, the archaea. They also look like bacteria, round blobby things under a microscope. Okay? That's all they look like, but genetically, these things are as different from bacteria as you are. They have a completely different evolutionary history. So basically, this is what he discovered, and what we're really going to be interested in is this root here, this root of this tree, which is the beginning of this tree of life, the root of this tree. So just to recap, what I told you at the beginning was this is what you see in the textbooks. That's the fairy tale. What you actually have is this. And if I draw this in a coarse-grained way, you can see that what's happening is you have an origin that branches into 2 and then branches into 2 again. And so these things are evolving in a simultaneous way rather than sequentially, as people had simply assumed. Now, there's an amazing surprise that they discovered when they did this work. This work was published in PNAS in 1977. And what they discovered was this. If they look at the root of the phylogenetic tree, which they could guess pretty much reasonably accurately even with their very first preliminary data, they found that it had to be at least 3 billion years old. Today, we know it's more like 3.8 billion years. Okay? So here's the thing. The Earth is 4.6 billion years old. The root of the phylogenetic tree of life is 3.8 billion years. So that means that we have a puzzle. Okay? Whoops. This is the one I wanted to show. The time available is short by comparison. Short, what did they mean? Well, you got from 4.6 billion to 3.8 billion years, that's less than 1 billion years. Life had to go from absolutely nothing, an abiotic planet, presumably, to something which is recognizably like the modern cell in less than 1 billion years. Very, very accelerated evolution. After that, evolution didn't do very much. Remember, everything you see on this tree of life was detected by looking at the ribosomal sequences and inferring their existence. So these cells that you see, even at the root of the phylogenetic tree, had an architecture similar, recognizably similar to the ones of every cell in your body. So, how is it possible that life went from nothing to such a complex cell in less than 1 billion years, probably a lot less than 1 billion years? And then after that, for the remaining 3.8 billion years, basically didn't do anything. Okay, maybe you had a few dinosaurs and humans and things like that, but evolution really didn't do much after that point. The structure of the cell was already decided 3.8 billion years ago, and that was a big puzzle. How could life evolve so fast? The other thing that they noticed, this is Carl Woese actually doing the mind-numbingly boring and painstaking annotation of various films in order to extract these data. This is his postdoc, George Fox, at the time. They were puzzled by this, and Woese wrote in 1977 that it is the mode of evolution that must have been different rather than the tempo. I want to explain what that means. People always assume that evolution proceeds in a certain way, and then to answer this paradox, you might say, okay, well, this evolution went faster at that early time. But that's not what I'm going to tell you, and it's not what Carl Woese guessed in 1977. What I'm going to tell you is that actually the way evolution proceeded in those early days was different from the way it proceeds now. It was a different mode, not just a different tempo. And there's a cosmological analog of it. If you look at the universe today, it started by, as far as we generally believe, a rapid process of inflation, and then followed by a power law growth, and rather than exponential growth of the matter and radiation-dominated universe, a much slower growth process. Biology has the same thing. I'm going to show you a very rapid exponential growth at the beginning, and then a transition to a much slower process of evolution, of expansion, if you will, and that's what we're going to see by looking at the genetic code. Now, in order for you to understand what that different mode is, I want to tell you how that mode of evolution is alive and well, and even today. So when you think about evolution, you generally think about millions of years timescales. I want to now, in the next couple of minutes, disabuse you of that notion. I want to tell you that evolution happened really, really fast, and you can even observe it happening in the laboratory. So this is a process called horizontal gene transfer, which is a process that's very fascinating to me as a statistical physicist, a condensed matter physicist. Let me tell you what it is. So here, these circles are meant to represent bacteria, and I'm telling you about horizontal gene transfer as opposed to vertical gene transfer. Now, vertical gene transfer is when you take your genes and you have children, you pass your genes on to your children, and then they pass their genes on to your grandchildren, and so on and so forth. The genes go down through the generations, and that's called vertical transfer. Horizontal transfer is something different. Horizontal transfer is when you transfer your genes to some organism that you're not related to. So here's an example. How many people here have seen the movie The Matrix? Oh, great, everybody has. Okay, fantastic. Okay, so you remember in that movie, there's a wonderful moment in it where the hero, Trinity, is stuck on a rooftop, and she's got to get out of a very, very dangerous situation, and the only way out is to fly a helicopter, which is conveniently there. However, there's only one problem: she doesn't know how to fly a helicopter. So no problem, she just phones up to mission control and says, beam the instructions to how to fly a helicopter into my brain. So they do that, and then she gets in the helicopter and flies away. Okay? Now that's science fiction, right? It can't happen like that. But if you were a bacterium, it could happen. It would happen. Well, I wouldn't have to teach my long and tedious courses on statistical mechanics that Chris Polly here in the audience has had to sit through many years ago. I would just be able to sort of spit out my DNA, and all of a sudden people would know how to do Feynman diagrams and 4 minus epsilon dimensions and things like this. Now, what? That would have been great. That would have been great, yeah. Yeah, it would have been great for me too. But I got paid to do it. So bacteria do this, and here's how they do it. So the one way they do it is they get invaded by a virus. The virus goes in, takes away the DNA from the host bacteria, makes lots of baby viruses. Those baby viruses take some of the host DNA, go and infect another bacteria, and sometimes they kill that bacteria, but sometimes they just give a gift of the DNA that they're carrying to the bacterium, and that way the genes are transferred from this bacterium to this one through the virus. There's other ways that they can do it. They can actually just sort of literally mate and swap DNA, and they can also just pick up DNA from dead bacteria in the environment. So biologists know this. As a sort of theoretical condensed matter physicist, I sort of think of this as the sort of, interaction Hamiltonian for the system. Now, the statistical mechanics question that you ask is this: if pairs of bacteria can do this, then what happens when a whole community does this? Okay, that's the statistical question. What is the system-wide consequence of horizontal gene transfer? So here's an example. So this is one that you've read about in the newspapers. It's a global health hazard. It's probably the largest health hazard we currently have. It's the spread of antibiotic resistance genes. It's much worse than HIV and various other things that you may have read about, Zika virus and so on. Because if this actually happens, if the bacteria evolve resistance to antibiotics, all of modern medicine goes out the window. You can't do surgery, et cetera, et cetera, et cetera. Okay, so how did this happen? The spread of antibiotic resistance genes happens because the bacteria in your gut are swapping genes like crazy, and some of them evolve genes that can confer resistance to antibiotics, and they swap those. And they swap those in the hospital environment, they swap them in your bodies, and those genes proliferate around the world. And if you create a new drug tomorrow, and you spend $10 billion to create a new drug tomorrow, in 5 years' time, resistance will start to appear, and in 10 years' time, the bacteria around the world will be resistant to that drug. And so we're seeing gradually various diseases that we've long been accustomed to solving by antibiotics are now becoming immune to antibiotics, and it's a major problem. Here's something that's a bit more interesting. This is a cyanophage. It's a bacteria. It's a virus that infects bacterium that lives in the ocean. It's a wonderful organism, one of my favorite organisms. It's responsible for most of the carbon fixation that goes in the oceans. As much carbon is fixed through the organism that this thing infects, a small bacterium, as all the croplands in the entire planet. And what's interesting about this is that the organism it infects is a cyanophage. It's a photosynthetic bacteria, a bacterium that lives by processing photons. Okay? But the interesting thing is that this phage, this virus that infects the bacteria, also has in its genome photosynthesis genes. And those genes get swapped back and forth between the virus and the bacteria, and they do it in such a way that they rapidly evolve the photosynthesis genes, and they cause the bacteria to be much better at processing light than they otherwise would be, and that's how this bacterium is able to expand its range throughout the oceans. And you get a collective state that arises because of the interaction of the viruses and the bacteria. I study this. It's a beautiful statistical mechanics problem. It's almost like the biological equivalent of superconductivity. So the take-home message is the cloud was invented 3 or 4 billion years ago by microbes, and not by CERN. And so there's a wonderful collective dynamics that goes on in the world of biology, and as a condensed matter physicist, that's what I'm interested in. Okay, so I've told you that evolution can be very rapid, so now I want to go through exactly how that happened. So the puzzle I want to solve now is life started 4.6 billion years ago. The last universal common ancestor, the root of that tree of life, was 3.8 billion years ago. That's not the beginning of life. That's the beginning of life was before then. That's the beginning of life as we can detect it. So what happened beforehand? And now I want to tell you a bit more about the genetic code. So there it is. I told you about it about half an hour ago. Let's look at the structure of it. The most hydrophobic amino acids are these ones. The most hydrophilic ones are these ones in blue here. So there's some kind of structure in this genetic code, and that was quantified by Carl Woese, who measured the counterplay between hydrophobicity and hydrophilicity. It doesn't matter how he did that, but for now, just know that the number that quantifies that, it's got a name, it's a horrible name, it's called polar requirement. It starts off small over here, it ends up big over here. So there is mathematical structure in this code, and we're going to use that structure to see that this code has a very interesting error minimizing property. So let's see how we would do that. So we've only got one genetic code, so how can we compare different genetic codes? So the idea is this: let's make a bunch of synthetic genetic codes in the computer. How do we do that? We just permute the labels. There's 20 amino acids, so there's 20 factorial different permutations. That's about 10 to the 18 possible codes. So we're going to use Monte Carlo simulation to sample these synthetic codes. So that's how we can create synthetic codes in a computer. The next thing we can do is try to see how good are those codes at minimizing errors. So how do we do that? Well, I'm going to roughly show you how one does that. And basically the idea is this: we're going to take that number associated with each code, with each amino acid. We're going to swap them round in that canonical genetic code table. And when we swap them round, we're going to add up the sum of the squares of the differences between the numbers of those amino acids. So we take the sum of the squares of the differences, add them up over all the code positions, and then we're going to get a figure of merit that tells you how good that genetic code is at minimizing errors. Because if it minimizes errors, then you should get amino acids that have numbers which are similar to each other, being close to each other on the genetic code table. And that means that this number, this figure of merit, should be small if the genetic code minimizes errors and large if it doesn't. So then what you do is this. You do your Monte Carlo simulation and you compute the probability distribution for that figure of merit for this polar requirement number. Now, here's what would happen if you had a frozen accident. Francis Crick's frozen accident would say that if you generated a bunch of codes at random, plotted their probability distribution, and then looked at the actual score, if it landed in the middle of this bell-shaped curve here, then you would conclude that indeed the genetic code is a frozen accident. It just arose by chance from what you would expect by random, and there's nothing special about it. But if, on the other hand, your bell-shaped curve looks like this, and your actual genetic code value is much, much smaller, say over here in the tails, then you would conclude that there is something funny going on, that this genetic code is not one that was chosen at random. And indeed, the actual experiment was done first most convincingly by Freeland and Hurst in 1998. Here's their results. Here's the bell-shaped curve, its frequency distribution for the figure of merit of how good the code is at minimizing errors. There's what you get at random. Here's the actual code value. The title of their paper was "The Genetic Code is 1 in a Million." Because in a million genetic codes, only one was this good. That was the actual genetic code. Okay, so that tells you then that the genetic code is special. It's not a frozen accident. It has evolved, and it minimizes not just it's not just unique, but it's also optimal. So how did it get that way? We're talking about codes, so I'm going to walk you through heuristically a kind of thought experiment which describes what was done in a mathematical and computer simulation of that mathematics in this paper here. So when we talk about codes, if I want to do a thought experiment, the thought experiment has to involve the experts in codes. So who are the experts in codes? Anybody? The NSA, exactly. These people, right? It's these people, okay? And anybody who didn't put their hand up when I asked about if you'd seen The Matrix, the people who didn't put their hands up are the people who know who these people are, okay? These are stars of 1960s and 1970s bad spy movies and TV shows. So what we're going to imagine now is a thought experiment where one of these spies is going to use a code, a regular code book, to communicate with another member of their fraternity, and we're going to see what can go wrong with codes and how biology figured out a way to fix this. So let's start off with our thought experiment. So Emma Peel here has a message, and she wants to transmit that to Austin Powers. So she transmits it, he gets out his code book, and he translates it, and it means this, and that's indeed the correct translation of this string of A, U's, and G's. But Austin Powers, of course, as you know, is a very careless individual. So this message, by the way, is unique. You know, you start off with here, you put it through the code book, you get the answer, the code works beautifully just as it's supposed to do. Being a careless individual, he's lost half his code book. And when he loses half his code book, the message doesn't make sense. It makes no sense to him, although it may make sense to some of you people. Okay? So what you learn from that is that if you have a deterministic code like this, like you're used to, if it evolves, if it changes, then the message gets completely garbled. That was exactly Francis Crick's point when he talked about the frozen accident. Okay? Codes can't evolve, because if they evolve, you get this happening and you read nonsense. Okay? So what's the solution? So the solution is you have a statistical code book. A code book that generates not just one message as an output, but an ensemble of different messages, and then one of that ensemble might make sense to you based on the context of the message, and then you say, okay, that must be what it is. So in this example here, we've got a probabilistic code book, and Mr. Powers has deduced that with probability 0.9, the message was this. Okay? So probabilistic codes are much more robust than deterministic codes because they're not so fragile. So let's see how life can evolve a genetic code. And to do that, I want to walk you through a simple thought experiment. So let's imagine the evolution not of life, but the evolution of automobiles. Okay, so the most popular car today in the United States, I think, is the Toyota Camry. Okay, it's a wonderful car, always works, its machines are very high precision, it's extremely reliable, it's very comfortable, and all of that. But if you went back 100 years, the cars that you would see were very simple cars. They didn't go very fast, they were bumpy, they always went wrong, and those sort of cars were certainly nothing like today's cars. But they worked, they got you from A to B. Now imagine that you were to drive over to your friend's house and the wheel comes off your car. Well, that's bad news because your friend has a Ford, you've got a Toyota, so there's nothing you can do. You can't fix it with, you can't sort of take his spare wheel and put it on your car or anything like that, or use any of his equipment to fix the car. You just have to go to the Toyota dealer. But back in the day, back 100 years ago, if you were driving one of these rickety old automobiles put together by talented inventors in their garages, well, if you went over to your friend's house and something went wrong with your car, you could just sort of get out some nuts and bolts from the basement and just sort of fix your car and everything would work. Okay? But you can't do that today. Okay? You can't do that with today's modern cars. I mean, you can't even build them in Detroit, so there's no way that you're going to be able to fix them in your own garage. Okay? So what we learn from this is that when you have a very simple organism or a very simple machine, its simplicity allows you to interchange parts, and they still work. But as the organism becomes more and more complex, becomes more and more demanding of the precision, in order for it to be able to work well, then the ability to swap parts is going to diminish. And so the idea then is that as the code evolves, it evolves along with life's complexity. As life becomes more and more complex, from rickety life forms that were like the old automobiles to modern life forms like us, which are like Toyota Camrys, if you will, life became less and less able to tolerate ambiguity, required more and more precision. And so what would have to happen was that life would co-evolve the code along with the complexity of life. And co-evolving a code means, in this case, that the code would have started off as being statistical, being basically, you get the right amino acids some of the time, but it doesn't really matter because the organism doesn't mind. Until today, if you don't get the right amino acids, everything goes pear-shaped. So you have this picture then of a distribution of proteins, statistical proteins, and that probability distribution gradually evolves to a delta function as you get to more and more complex life. So let's see how we can take that picture and turn it into a theory. So let's start off with this question. Selection pressure operates on the organism. It doesn't operate on the code at all. It operates on the organism. Selection pressure means if you can reproduce more, you will win in the population. That's what natural selection means. So here's an example of it. So I'm a physicist. I like to write papers with tech, as most of you do. But when I write grant proposals, say with biologists, they like to use Word. So let's imagine a scenario where the National Science Foundation, or maybe the Department of Energy, says, I want proposals in some area of biology, but it has to be quantitative. Okay, you can't just measure something and write a story about it. You've actually got to have a mathematical scientist produce some quantitative analysis of it. So your biologist friend goes and finds a physicist to work with, and you decide you've got a good idea, and you start to write the proposal. But the problem is I write my part of the proposal in tech, my friend writes her part of the proposal in Word, and we keep sending out each other's files back and forth, and I use Unix, so I don't know what Word is, and she doesn't know what / means in tech and so on. And so we end up never actually submitting the proposal. Meanwhile, my colleague down the hallway, who's had a bit more experience, just says, okay, never mind, we'll just use Microsoft Word and go with it, and they produce the proposal. Even if it's not as good a proposal, it still gets accepted by the NSF by the deadline and gets funded. So next time round, of course, you write your proposal in Word. This isn't a hypothetical scenario, by the way. This is actually, has actually happened. Okay? Okay, so selection pressure can influence the choice of the code that you use. Okay, now here's another thing. Let's look at different innovation-sharing protocols. Protocols are like operating systems. So here's two operating systems. Here's Bill Gates with his Windows computers. Here's Steve Jobs, the late Steve Jobs, with an early example of a Mac computer. And of course, you all know that something like 95%, 96% of the world's computers run Windows. A very small percentage actually run the Apple operating system. And why is that? Well, you know, I've got a Windows machine, and I can tell you what the reason is. The reason is that the Windows computers are better. They're much better. They're much more stable. That's a very inopportune time to have a blue screen of death. Yeah, the Windows computers are great. They are stable. They work. They always never go wrong. Yeah. But, okay, they have some problems, yes. Windows computers do have problems compared to Apple computers, but there is an advantage of Windows computers, and there's a reason why people use them. And don't worry, the talk hasn't just crashed and burned. It's just a joke. The reason is this. The reason why you use Windows, or most people do, most businesses do, and most organizations do, is because there's more programs written in Windows than there are for the Mac operating system. Okay? It may not be true today, but it certainly was true for most of the history of computing, and it's because Microsoft made its software development kit available for free to all developers to use. Apple isn't so nice, and so people just wrote programs for Windows, and then once more and more people were using Windows, if you're going to write a program, you should write it for Windows because there's going to be more people who'll buy it, and so it's an autocatalytic effect. In fact, this came so bad that Steve Jobs actually changed the chip that Macs used to be the same chip as Windows computers use, in order that you could run Windows programs on Macs. And in fact, today, the lesson is very well learned. What was the big advantage of the iPhone? What was the big advantage, apart from the fact that it actually worked? Apps, the App Store, okay, 1 million apps on the App Store, only 100,000 on the Google Store. And, well, now it's the same, but for many years it wasn't. So we did a computer calculation. So here's the calculation, and what it is this: we did a digital life simulation. We simulated life that was artificial life inside a computer, kind of like a computer virus, but benign. Digital organisms that do the following: they're asexual. They evolve and they reproduce. They have a phenotype, which is a function of their proteins. They have a fitness, which depends on what their proteins are, and that enables them to reproduce. The proteins are obtained by translating a genome with some genetic code, but with errors. And the individual reproduction rate is a function of fitness. So all the key universal ingredients of living organisms are represented in this community of co-evolving computer viruses. And then they just sit inside the computer and they fight amongst themselves. And what we're trying to understand is this: if I've got a whole community, each of which has many different genetic codes, what's going to happen to the system? So here's what we measured. We measured how optimal the system is. We wanted to see how good are the codes in this community, in this putative community, at minimizing errors. And so we measured the average amino acid distance between neighboring codons. We looked at a distance metric, and we could see how good the different amino acids were if they were adjacent to each other in genetic code at minimizing errors. Then the other thing we did was we counted how many codes are present in the population and measured their average distance in some measure. So here's what you get when you do this. So here is the quality of the genetic code measured in some way. I'm not going to go into it in great detail. And this is time. We did an experiment which has a control. The control is this: we're going to do regular Darwinian vertical evolution of our organisms, and what we're going to see is this is shown in red. What happens is the codes start off here, they have some average distance, and the codes go down so the distance gets smaller. Smaller means that the code is getting better at minimizing errors. But then what happens is the code stops, and then it just is a constant up to here. And then you do it again, and the measurement goes down like this, and then it comes along like this. You do it again, and you get this. These lines don't evolve in time. This, in fact, is Francis Crick's frozen accident. What happened here is whatever genetic code you have is just whenever happens to be the time when this code evolution froze. And if you do a simulation here of the distribution of random codes, the Monte Carlo simulation I told you about earlier, you get a random distribution, a bell-shaped curve that looks like this. These codes are just what you would get from random. That's with regular vertical Darwinian evolution. Nothing we could do could stop these codes from getting trapped in local minima and getting frozen. So then we said, okay, fine, let's do something different. Let's switch on horizontal gene transfer. Once you switch on horizontal gene transfer, even a little bit, its story is different. What happens is the codes keep getting better and better and better, and eventually asymptote out until you get a very high quality code down here, which is very, very different from what you would get at random. And so what you learn from this comparison is that horizontal gene transfer is what leads to optimal genetic codes. Similarly, if you look at the number of codes and the distance between the codes, you start off with a community with 40 initially random codes, all fighting amongst each other to see what happens. What happens is that in red is with vertical Darwinian evolution, you get frozen accidents and something kind of like what you would get at random. When you switch on horizontal gene transfer, you go down to a state where there's only one genetic code. So what we learn from this then is horizontal gene transfer leads to universality. So here's what I've told you. Okay, so this is now the interpretation of this, and this is now what I've told you up to now has been purely based on data and based on simulation. So now let's put it all together. So here's what I've told you in this talk. I've told you about the bacteria, the archaea, and the eukaryotes, the three domains of life that Carl Woese discovered. This is our cloud of ignorance surrounding the last universal common ancestor. What I've told you is this. Prior to this period of time here, this is about 3.8 billion years, we looked at what would happen if you had a state where you had many different organisms with different types of genetic code all competing amongst each other. You have ambiguity because you've got very simple organisms. They can swap parts, they can swap genes, and it still works. But you can only swap genes with organisms that have the same genetic code as you do. There's no global horizontal gene transfer. And what I showed you was, by argument and by showing you the graphs from our simulations, was that eventually you get to a point where you get an emergence of a universal global genetic code. You have a first universal common ancestor. At this point now, you have a very interesting situation. You have a universal genetic code, plus you've got very, very simple organisms that don't mind if they take in genes from other organisms and swap genes around and so on. So you have a universal code with a toleration of ambiguity, a toleration of alien DNA. That leads to a communal state with an explosive growth in innovation sharing. Think of the internet, 1993 onwards. Okay, what happened then? Hypertext Transfer Protocol was implemented on top of TCP/IP. Suddenly, all the computers in the world were able to talk to one another and transfer data. Bang! You started the internet. Explosive growth that led to today's internet. So, of course, this ends. There's a period of time where I'm not going to talk about it. There's a transition which we call the Darwinian transition, where in fact the organisms become so complex that they can no longer tolerate swapping of parts, just like the Toyota Camry to today's automobiles, and you make a transition to this era of vertical evolution. This era of horizontal evolution is the progenote. It's an era where there is no notion of phylogeny, because genes aren't transferred to descendants, they're transferred to everyone. You don't have a tree, you have a reticulate evolution. You have a network. It's not the individual lineage that varies in descent, as Darwin wrote, it's the community that varies in descent. It's a collective state of life. And it's the only way we know to explain the properties of the genetic code. So I'm not going to go through this, so there's that. That's all for another talk. Let me wrap up. What I've told you is that the world started in 4.6 billion years. The vertical evolution starts about 3.8 billion years. And somewhere between here and here, you have an era of collective evolution, where horizontal gene transfer on a massive scale, where organisms and species, individual species don't exist because there's no notion of tree, there's no notion of family tree. So you have a network, a collective state. The collective state uses the network effect to evolve rapidly and doesn't get trapped in local minima, and that led to the root of the phylogenetic tree, where the regular population genetics theory of evolution took over. So that's the end of my talk. So thank you very much. I'll be happy to take questions.
So please wait for the microphone for questions. I go up here to.
Can you just in 1 minute explain how homochirality appeared?
Sure. Let's see. How long have you got? I don't have real time to explain this, I'm afraid. Okay, it's a very interesting story. The short answer is that homochirality arises through a mechanism which is purely based on autocatalysis. Okay, if I had more time, I would be able to talk about it, but I can't get it all into 1 hour talk. So what we've learned is this: homochirality. The question was, how does homochirality arise? What is homochirality? Homochirality is the fact that every amino acid in your body and every living organism is left-handed. It breaks chiral symmetry 100%. Okay, and it's a symmetry which is kind of like baryon asymmetry in the universe. And the mechanism that we discovered is a stochastic mechanism for amplification of asymmetries in chirality through random noise. It turned out to be multiplicative noise, and it's not based on symmetry breaking as you would normally expect. It's based on amplification of noise, and I can't explain it, but it ends up being that the system, it will, even if it starts from a symmetric state, will break chiral symmetry 100% along enough times. And I think it's interesting because I wonder if there's autocatalytic mechanisms for baryon production, and so it may be that this mechanism that we've discovered is related to maybe something cosmological. So I can't explain it to you in detail now. But if you want to read about it, there's a really nice physical review letter somewhere here. So this one. Okay, next question.
So based on what you know and what's being learned from these sorts of approaches, what would you say a reasonable estimate is for what fraction of physical environments and chemical environments similar to early Earth that would, within 1 billion years or so, develop something we would call active life?
So I don't know, I can't give you a scientific answer to that question, but for several reasons. One is that nobody agrees upon what the early environment of Earth was like. Secondly, that does involve details like chemistry and so on, which I'm not qualified to say. But I will say this, I'll tell you what my prejudice is. My prejudice is that in any environment, life will evolve. Okay? My prejudice is that life is an inevitable consequence of the laws of physics, that if you have systems that are sufficiently far from equilibrium, they will form aggregates which are hierarchically organized with multiple dynamical levels of feedback between them, and eventually eventually evolvability will evolve, and at that point the system will be able to evolve in an open-ended way a complexity. I know how I can make simple models of how complexity evolves. I don't have a simple model for how life started. The best candidate, according to some people, for where life started is a hydrothermal vent under the ocean, but not the usual hydrothermal vents that you see on Nova, but alkaline ones, not the black smokers, but the warm alkaline hydrothermal vents such as Lost City. And the reason that people think that this would be the good place is because of a geological process called serpentinization. And serpentinization is interesting because it produces proton gradients which are in the same direction as the proton gradients in all living cells. And so and that gradient direction, by the way, is the opposite way to the way you would have designed it if you were trying to do intelligent design on it. So people think, aha, maybe this weirdness of all cells reflects the initial conditions under which life evolved, so maybe life evolved under that environment. Here's the good news: serpentinization also exists on Mars in spades, and may also exist on Enceladus, the moon of Saturn, where we now know that there is a global ocean underneath the crust of the planet, and we can even sample the water that comes out of that planet, out of that moon, and so we might be able to detect life arising through these mechanisms in the next 10, 20 years. So NASA is very interested in these sorts of mechanisms, and our institute at Illinois is working on that too. So I hope that's the best answer I can give to your question. But I will tell you this: everywhere we look on Earth, we look at an environment, we say, this environment won't be able to support life. We find microbes living there, even in rocks kilometers below the ocean floor. Yeah.
So towards the beginning, you took a poll about the uniqueness of the code. And
Yes.
in your talk, you said that our code is 1 in a million, but there's many more than a million codes.
Oh yes. Well,
So the question, are all the highly optimal codes very near to each other in some sense?
our code is not optimal, and it doesn't have to be optimal. Okay. In biology, you don't have to be the best. You just have to be better than the other person behind you, right? So if you're being chased by a bear, you don't have to outrun the bear. You just have to outrun your hunting colleague. Okay, yes, there's 10 to the 21 codes. Now, you can construct codes that are more optimal than the one that we have. That's true, but it's good enough. And see, here's my answer to the question. You can see here that I do this experiment many, many times. Each of these trajectories is a different rebooting of life, and they all emerge at the same dynamical fixed point. When I do it with vertical evolution, you get trapped in local minima, and so you get a non-unique final state.
So every one of those tracks converge to exactly the same code?
Yeah. Very, 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 very within our ability to measure, yes.
Okay, and the second question has to do with the 20 amino acids.
Yes.
Are they also optimal?
No. I don't know. I doubt it. So there's some questions that you can ask physicists and expect a reasonable answer, and some that you can't. If you ask a physicist, can you explain to me superconductivity? The answer is yes, I can explain to that. Okay, I know the BCS many-body theory explanation of superconductivity, or I know how to make a quark condensate, or something like this. If you ask me, calculate the critical temperature of a superconductor, I can't calculate that because it depends on microscopic details. So if it depends only on coarse-grained details, universal properties, then we can answer the question. If it doesn't, then physicists can't answer it. In my opinion, why there are 20 amino acids is one of those questions that is like the critical temperature of a superconductor. It depends on details, it depends on historical contingency, and there's no rhyme or reason why it should be 20 or 21 or 19. We can even make in the lab artificial systems which have different numbers of amino acids in them. There's nothing fundamental about the number 20. And in one other version of this talk, I explain a beautiful paper by Francis Crick, where he came up with a wonderful number theory explanation for why there's 20, and even predicted what the codes would be. This was before the genetic code was actually discovered, measured, and it's completely wrong. It predicts the right number, but the actual genetic code is not one of the codes that he predicted could be there. So no, I don't think we can understand that without understanding more chemistry than I know. There's other people who claim that they understand these things, and I have no reason to disbelieve them, but it's not what we're interested in here in this talk is the dynamical processes of evolution.
Yeah, so it's not clear to me why complex entity is less likely to benefit from horizontal gene transfer. You know,
Right.
if I imagine writing a computer program,
Yeah.
I want to take advantage of modular design and
Yes.
collaborate with my colleague. You know, if there's something that prevents me from collaborating with my colleague, you know, maybe then we would develop codes in separate directions.
Right. So the question was, why can't complex systems still do horizontal gene transfer? Well, I think the reason is this. We actually did a calculation of this, a model calculation, which I didn't have time to show you. But what happens is that eventually the likelihood that as you get more and more optimal, the likelihood that you're going to get a gene transfer that does something that you don't already do is minimal. And so there's no benefits to having the horizontal gene transfer. You're much more likely to get something that's deleterious than something that is good. So you'd put up a firewall around it, like you have a firewall around Fermilab's website, computer systems and so on. So systems put up firewalls, and firewalls emerge naturally because even if you have horizontal gene transfer going on, you're transferring most of the time the same genes between things, so it doesn't have any effect. So that's what happens in complex systems. One more. Yeah.
You've given us a window after the 4.6 billion years ago point, during which all the processes that lead to life have to arise. And you marked a timeline with a few of the events
Right.
that have to go in there. And I gather we don't know a whole lot about when some of those things came in. And I wonder if you could sketch out what hope there might be for establishing kind of a clock. I mean, all of the processes of life might have evolved in the last 100 million years instead of taking the whole 800 million years. Or, the community of people who try to figure out how life started do they have a hope of establishing some boundaries on this in the future?
They do. So the question was, can we put a timeline on this early phase of life? So you can do the same kind of thing that you do in archaeology. In archaeology, if you find some bones in some rock layer, and you know the age of the rock layer by radioactive decay or something like that, then you know the age of the bones or the pottery or whatever it might be. So you can start doing these kinds of analyses where you look at complex early versions of proteins or like the ribosome or other machinery in the cell. And if you can date some parts of it, but they're embedded with other parts, then those other parts can then be dated, and you can ask how they got that way. So people are starting to do this kind of stratigraphy. Exactly, that's the word I was looking for. Thank you. The stratigraphy of early evolution on parts of the translational machinery and so on. People have just started doing this. The work I know about is unpublished. It's that new, but there is hope that we can make timelines for these things, and I can't tell you the evidence for this, but the idea that this polar requirement is the number that tells you about the evolution of life, that turns out to be, until very recently, I never really knew a good reason why that number should be the one to use. But now I do, from looking at these kinds of studies. So yes, we have opened up a window on early life and the evolution and the time scale of it by looking at things like the genetic code. Thanks for the question.
So it's getting rather late. Why don't we thank our speaker again? And